tracking glucose carbons. We introduced this idea last time we wrapped up with this slide, so I'll elaborate a little more on this slide and tell you about some of the tricks and isotopic gymnastics that we can do in trying to track labels through glycolysis. Then we're going to move on to the next reaction. It's going to be the first reaction in the so-called payoff phase of glycolysis. She's waving at me. You can't hear me. The mic is on, so I'm not sure. I've got a green light. I'm not talking to myself. There's a person in the booth talking to me. Okay, well, I'm going to go ahead and go, and she can sort that out. So then we're going to go ahead and move forward with GAPDH, which is a reaction that is going to be the first reaction in the so-called payoff phase of glycolysis. Remember, that's when we're going to reap the benefits of the ATP that we invested in the early stages of glycolysis. Then what we're going to do is we're going to talk about a specific experimental design called the isotope exchange experiment. And that's going to tell us about the nature of the GAPDH reaction. Okay, the isotope exchange experiment is going to tell us about the nature of the GAPDH reaction, and I'll elaborate more on the isotope exchange experiment, but this is something that I want you to make sure and learn. We've already mentioned it initially in the context of complex 5 when we said that we isolated complex 5, and I'll elaborate more on that. And really today try to paint a more clear picture of what that isotope exchange experiment is, and hopefully in the context of GAPDH will give you a sense of how to apply it to different types of situations. And then we'll wrap up today, I hope, by getting to phosphoglycerate mutase, which is going to be another enzyme involved in the isomerization of different units in glycolysis. So here we were tracking carbons in glycolysis, and we said that we ended up making, after this aldolase reaction, we ended up making 2 trioses. And the interesting thing about the trioses, remember we worked pretty hard in glycolysis to try to get some symmetry. And although we got a lot of symmetry, and that allowed us to generate 2 3-carbon units, the 2 3-carbon units are not identical. Right? The 2 3-carbon units aren't identical because we've got the dihydroxyacetone phosphate and the glyceraldehyde 3-phosphate. Okay, so to close the loop and make sure that we have the same thing, remember the word I said was economical, we want to make sure that we can use all the same enzymes for the back half of glycolysis as we used in the first half. And to try to achieve that, we are going to use this triose phosphate isomerase reaction to be able to shift those back and forth. And the reason I show you this line, this slide right here, is because it allows you to see that the 1, 2, 3 carbons of fructose 1,6 bisphosphate end up generating the carbons on dihydroxyacetone phosphate, while the bottom 4, 5, and 6 carbons end up generating the glyceraldehyde 3-phosphate molecule. Okay? And I'm going to elaborate on this slightly with another picture here on the board to try to bring this home a little more, because this is something that I do find is sometimes tricky for students. I guess I shouldn't start on this board. I should start on this board. But what I'm going to do is I'm going to tell you about, and this is highly redundant with the slide that's currently up there, but I'm going to give you a picture from glucose carbons. So where do these things come from in terms of the glucose carbon number? And I'm going to go ahead and draw on here the glyceraldehyde 3-phosphate molecule. So this is the same molecule that we're going to produce at the end, after that, the triose phosphate isomerase reaction. We end up generating 2 of these. And we're going to number these by priority. And remember that we can represent the phosphoryl groups with a P and a circle around it. And we're going to number these by priority with the 1, 2, and 3 carbons as shown here. And recall that this glyceraldehyde 3-phosphate will then go on to all subsequent reactions in glycolysis. So all subsequent reactions in glycolysis are going to use this. And we want to be able to think about what carbon numbers this comes from. And so if we have the 1 carbon label, and you ought to be able to extract it actually from this slide here. Okay, but it's a little confusing here with this slide, and so all that this picture that I'm drawing on the board offers in comparison to the picture on the slide here is that here I've only shown you 1 glyceraldehyde 3-phosphate molecule and the numbering for dihydroxyacetone phosphate. But remember, we're going to end up with 2 molecules of glyceraldehyde 3-phosphate, and it's straightforward to see how that gets converted, but if you don't see it, we'll draw it here on the board, but recognize that this gets flipped. Okay, this gets flipped. It's drawn here on the slide to show you how it's oriented within the fructose 1,6-bis phosphate molecule. Okay, namely that phosphate is on top here, but that phosphate, when we do the interconversion, ends up being down here. Okay, so that's why it gets flipped. So in that way, the 1 carbon here on glyceraldehyde 3-phosphate is going to come from the 4 or the 3 carbon of the glucose molecule. Okay, so hopefully that's clear. It's not quite aligned. But this 4 or 3 carbon is going to label the 1 carbon in glyceraldehyde 3-phosphate. Okay, the 4 or the 3. That means if we label carbon 3 in glucose, okay, and then and glycolysis is the only thing producing glyceraldehyde, that means that 50% of the glyceraldehyde will be labeled. Why only 50 at the 1 position? Why only 50%? Well, because the other carbon is going to be coming from the 4 carbon. If we only label 3 carbon, then it's not going to have a label. Okay, so that's the kinds of exercises we can do. The second carbon is going to come from the 5 or the 2nd carbon of glucose. And the last carbon here, carbon 3, is then going to come from the 6 or the 1 carbon from glucose. Okay? So one of the things that you can think about, if you label the 1, 2, or the 3 carbon in glucose, okay, these are the kind of tricks we can play. So this is just to demonstrate the way we might play with these labelings to try to appreciate what's happening with glycolysis. So imagine a situation where you label the 1, 2, and 3 carbons of glucose. Okay, the 1, 2, and 3 carbons of glucose. Yeah, I'm not sure why it's not on. It's got a green light. So. We're going to label the 1, 2, and 3 carbons. And now let's imagine that we block triose phosphate isomerase. Okay, we block triose phosphate isomerase. Now, interestingly, because remember we can still generate glyceraldehyde 3-phosphate from a previous reaction, we're still going to have some glyceraldehyde 3-phosphate because the aldolate is producing it. So that can go on and do the rest of its business in glycolysis. Right? But the dihydroxyacetone phosphate cannot. Okay, so it cannot if you block that triose phosphate isomerase. So if you only label these carbons here, then nothing downstream of glyceraldehyde 3-phosphate can be labeled. Okay, so that's the kind of gymnastics that we can try to do. Okay, so what I'd like to now do is tell you about the next reaction. And again, that's going to be the first reaction in the payoff phase of glycolysis. So we've crossed that blue-red line there, that threshold, which means we are in the second half. Okay, that's the way we've designated glycolysis. The payoff phase is everything at the beginning, and the investment phase is everything in the beginning, and the payoff phase is everything at the end. Okay, and in that reaction there is going to be glyceraldehyde 3-phosphate. And glyceraldehyde 3-phosphate is a really important reaction. The reason it's important, well, first of all, from a mechanistic perspective, it's probably one of the more interesting enzymes in glycolysis. But the other reason that you can argue that it's very important, one of the more important reactions of glycolysis, is because it involves that NAD plus and NADH. I've told you already numerous times that a key aspect of glycolysis, in fact, its dependence on oxygen, arises because of that NAD plus, that NADH there. Okay, and this is the oxidation reaction, and the reason that glycolysis interfaces with oxygen. This is the reason that glycolysis is connected to the oxygen reaction. So let me go ahead and show you that reaction. First, before we do that, maybe I'll give you a little bit of a perspective of where we're going in glycolysis. Okay, a little bit of a perspective of where we're going. So we're going to have 2 molecules of glyceraldehyde 3-phosphate, perfect symmetry. Worked hard to create 2 things. We've got redundancy. That's economical. After that, we're going to do our glyceraldehyde 3-phosphate reaction. I'm going to talk about that most of today. And then we're going to generate the product, which is 1,3-bis phosphoglycerate. Okay, 1,3-bis phosphoglycerate. Notice that that product now has 2 phosphates. The product now has 2 phosphates. And that sets us up in a really great position, because what we're going to do in the next reaction is we're going to immediately remove one of those phosphates. We're immediately going to remove one of those phosphates. And where are we going to put it? Well, it turns out we're going to put it on an ADP. Okay, we're going to pull it off and put it on an ADP, and that allows us to make ATP. Okay? And that allows us to make ATP. Then what we're going to do is we have 3-phosphoglycerate. We're going to do another isomerization reaction to try to move that phospho group, that phosphoryl group, from the 3 position to a 2 position. Okay, we're going to move it from the 3 position to a 2 position. And you might ask, why are you going to move that now? You told us up here that the goal was to try to get symmetry, and that's why you were rearranging things. But why are you going to take the phosphoryl group from the 3 position and put it on the 2 position? How does that add to any of the symmetry arguments that you made earlier? Well, the reason we do that is because what happens next is very chemically elegant, very smart of glycolysis, because after we move that phosphoryl group from the 3 position to a 2 position, what we then do is we pull a water out of that triose. Okay, we're going to yank a water out of that triose. And what that ends up doing is it creates an enol that cannot tautomerize to its keto form. It creates an enol that cannot tautomerize to its keto form. The reason for that is because it's stuck, it can't tautomerize, because the phosphate that we left on there at the 2 position prevents it from doing so. The phosphate will not let it tautomerize, and it really, really, really wants to tautomerize. Okay? And as a result, when we move that phosphoryl group off, when we pull it off, it says, finally, I can tautomerize, and when it does so, it releases a lot more energy. Okay, so by moving the 3-phosphoryl group from, by moving the phosphoryl group from the 3 position to the 2 position, we set ourselves up to get a lot of energy a little later on in glycolysis. Okay, so let's now take a look at the GAP-DH reaction. Okay, sorry. So let's go ahead and take a look at the GAP-DH reaction. And here it is. And you'll notice that what we're doing here is, as I've said, we're doing a phosphorylation. We're adding the phosphate on. The phosphate's being added to that position there, the 1 position. Okay, it's being added to the 1 position. And the thing that I want you to star and underline on your slide is that this is not a kinase reaction. This is not a kinase. Okay, it's a little peculiar because we're phosphorylating the triose. Okay, and every other time when we've talked about phosphorylation, we've done that phosphorylation to date by using something like an ATP. Okay, notice here that's not how it's happening. We're actually taking the inorganic phosphate in solution and adding it on to the molecule. That should look strange to you because you haven't seen that before. Right? That should look a little peculiar to you, and I'm going to explain how that happens. Notice that the delta G naught prime of this reaction is about 6.3 kilojoules per mole. Okay, so a little positive, and I'll explain the nature of that delta G naught prime here momentarily as well. Okay, so this reaction does a couple of different things. It contains 2 different processes.
This one will work if you'll
Is it recording?
You can hear this.
Okay. Sure.
And we'll fix this one before next class.
So what we have here then is we're going to make this compound here, okay, that has the 1,3-bis-phosphoglycerate, and we're doing 2 things. And one of the things that we're doing is we're doing an oxidation reaction. Okay, that's one thing that we're going to be doing. And the second thing that we're doing is we're forming an acyl phosphate. The second thing that we're doing is forming an acyl phosphate. So let me tell you about acyl phosphates. This is a new kind of compound that we haven't seen yet. An acyl phosphate, so I'm going to tell you about acyl phosphate here. An acyl phosphate is the hydrolysis is going to yield a ton of energy. Okay, acyl phosphate bond hydrolysis is going to yield 49.3 kilojoules per mole of energy. Okay, so what is an acyl phosphate? Well, an acyl phosphate is when you have something like a carboxylic acid. So you've got an R group down here, whatever it is. You have a carbon double bonded to an oxygen. And then here you have a phosphoryl group. Okay, there you have a phosphoryl group. So it's just a carboxylic acid where you've replaced the H on the OH, on the alcohol, with a P bonded to 3 oxygens. Okay, that's an acyl phosphate. And the acyl phosphate, as it turns out, is going to have a really high energy. Its hydrolysis yields a lot of energy. Its hydrolysis yields a lot of energy. For actually the same reasons that cleavage of a phosphoanhydride bond, hydrolysis of a phosphoanhydride bond, yields a lot of energy. It's the same resonance arguments that we made there. It's 49.3. It's good. It's not 493 good. It's 49.3 good. So it is the same reason. So if you go back and look at the arguments that we talked about for ATP, you can see that some of those same things will apply. So acyl phosphate hydrolysis is what we're talking about here. And when we talk about glycolysis, one of the terms that I've used throughout the semester and that you've heard already me say a couple times, but I haven't really distinctly defined, is a term called phosphoryl transfer potential. A term called phosphoryl transfer potential. Now, phosphoryl transfer potential gets used extensively in glycolysis, okay, less so in the TCA cycle. The reason it gets used extensively in glycolysis is because, remember, I've told you already that all of the intermediates in glycolysis have phosphates. All of the intermediates in glycolysis have phosphates. So we like to think about the bond energy of those, the energy of hydrolyzing those phosphates, of pulling them off, and thinking about that in terms of phosphoryl transfer potential allows us to do that. This is also a term that the MCAT likes to use, and so I want to make sure that it is in your lexicon. Okay, so phosphoryl transfer potential is simply the amount of energy that's released when a phosphorylated compound transfers its phosphoryl group to water. Okay, you've got a phosphorylated compound, and when you take the phosphoryl group off and put it on water, that's the phosphoryl transfer potential, the amount of energy that's released in that process. Okay, and really the reason we use this is because it allows us to compare everything to ATP hydrolysis. It's a really good metric to stack everything up against ATP hydrolysis. So, for example, when we talk about this product of the GAP-DH reaction, okay, which is, I've told you already, is an acyl phosphate, it's 1,3-bis-phosphoglycerate, it has a higher phosphoryl transfer potential than ATP. Right? ATP has something like a negative 31 kilojoules per mole, and this compound here has actually quite a lot more. Right? It has quite a lot more. This acyl phosphate, the 1,3-bis-phosphoglycerate that's produced in this GAP-DH has a really high phosphoryl transfer potential. It's kind of amazing. And I also want you to recognize that GAP-DH is producing the first high-energy intermediate in glycolysis. So let me tell you a little more about the GAP-DH. And that first high-energy intermediate is the 1,3-bis-phosphoglycerate. I told you that GAP-DH is really catalyzing two different types of things. The first thing that it's catalyzing is an oxidation reaction, and the second thing that it's catalyzing is a acyl phosphate formation. And so we can think of the GAP-DH reaction as really catalyzing two different processes. Really, GAP-DH can be thought of as catalyzing two different processes. And the first process is going to be the oxidation, and the second one is going to be the acyl phosphate formation. So I'm going to draw each of those out for you. Okay, so let's talk about the first one. And what I'm going to do here in showing you this first reaction is I'm going to give you the substrate here, and I'm going to add a deuterium on it. I'm going to represent that deuterium as an H with a superscript 2. And the reason that I do that is because we're going to show here in a second that we've got an NAD involved, NADH involved, and I want to avoid the awkward NADD. Okay, and what we're going to have here then is we're going to have NAD plus and H2O. Okay, and what we're going to do is so this is our glyceraldehyde 3-phosphate. There's the aldehyde, that's the phosphate. This is the substrate for the GAP-DH reaction. This is our starting point. Okay, and this is going to be the first process that you can think about GAP-DH catalyzing. All I've done here is add that deuterium. I've added the NAD+. Remember that the NAD+ is what's required. We need the NAD+ for the reaction, so I've already added that NAD+. I haven't added yet the inorganic phosphate because the inorganic phosphate is going to be involved with the second process I'm going to tell you about. Okay, and we've also added a water here. Now we're going to do an oxidation reaction. And as it turns out, that oxidation reaction, I'll label it here for you, that oxidation reaction is going to be extremely favorable. So I'm drawing the reaction arrow with a very large arrow, long arrow pointing in the forward direction, and only a tiny little small arrow going in the backwards direction to try to indicate that that is positive. And what we're going to do here is we're going to oxidize that species, that aldehyde. Okay, so what we're going to end up making here is that carboxylic acid, as shown. And then we're going to end up with everything else is going to be the same here. So I'm just drawing the same part of the compound over here. But what's notable is that we form that deuterated NADH. Okay, so it's the NADH with the NADH with that H superscript 2. Okay, why? Because it was a direct hydride transfer. Okay, that is a hydride transfer, the same type of stuff that we were talking about earlier when we showed the reaction mechanism that I keep talking about early on in the semester for lactate dehydrogenase. We've got the same sort of spirit happening here, where we've got that hydride, and now it's ending up back on that NAD here in the product. Okay, and the delta G naught prime for this reaction is really impressive. You're going to really be excited when you see this. It's negative 50. Negative 50. So that oxidation reaction is a very favorable reaction. Okay, at a negative 50. Okay, so that's the first process that's going on by the GAPDH. Now what I'd like to do is tell you about the second process, because that's only half of what GAPDH has to do. Oxidation's one part. We've taken care of the oxidation, we've taken care of the NAD plus and the NADH, but there's a second part of this, a second component, the GAPDH, and that is going to be the acyl phosphate formation. So I'm going to now represent what's happening in that reaction in a separate reaction drawn on the board. That process, I should say, with the second reaction here on the board. So now I'm drawing the product of that. So I drew that first process, and now I'm taking the product of that first process, and I'm adding, I'm going to start with that, and I'm going to add now, I told you that I did not add the phosphate, the inorganic phosphate in the first process. Now I'm going to add it in the second process because it's going to be important. And what I'm going to do in the second process, much like I label with here a deuterium, an H superscript 2, much like I labeled a deuterium in the first process, I'm going to label that inorganic phosphate, so P superscript 32, radioactive phosphorus there, I'm going to label that in the second reaction. Okay, so we've got the product of the first process. This is exactly the same as that. Okay, but now we're adding the inorganic phosphate, and the inorganic phosphate then is going to react, and much like exactly the opposite of what I showed you above, we're going to have a very unfavorable reaction. So this is an acyl phosphate formation. This is the acyl phosphate formation part of the process or reaction. Now the acyl phosphate is the same thing that I told you over here, which has a big hydrolysis, a big free energy of hydrolysis, okay, a big negative phosphorotransfer potential. And this is going to produce a species that has that acyl phosphate. So here it is with the, sometimes we draw that with a P circle, but you have to recognize here that it's P with 4 oxygens attached to it. And now we have the rest of the molecule, which is going to be the same. And notice that that phosphate, because this is a phosphorylation reaction, even though it's a phosphorylation even though it's not a kinase, notice that that inorganic phosphate does get attached to the molecule, and then we end up with our H2O over here. And this delta G naught prime, coincidentally, is about positive 50 kilojoules per mole. Okay, so these two reactions, the first reaction is exergonic to about the same extent that the second reaction is endergonic. Okay, so this is very unfavorable, very endergonic. It really wants to go backwards, whereas the first reaction really wants to go forward. Okay, so let's think about these two different reactions then, and let's draw, we talked earlier in the semester about drawing reaction coordinate diagrams, so let's draw a reaction coordinate diagram if we were to conduct each of these two processes separately. Okay, let's draw a reaction coordinate diagram if we were to conduct these two processes similarly. So this is going to be my reaction coordinate diagram. Remember, this is going to be, the y-axis here is going to be free energy. Okay, we've drawn this before earlier in the semester. It's been a while, but you've seen this before. This is how we introduce the concept of exergonic and endergonic when we first start talking about delta G. And what we're going to do there on the x-axis is we're going to make that reaction progress, and this is going to be free energy on the y-axis. Okay, and I should tell you that this is hypothetical because I don't want you getting confused that this is the way that GAPDH works. This is a hypothetical, assuming that each of those two processes occur as separate, distinct reactions. Okay? Hypothetical, assuming that these two processes that I just showed you, numbered 1 and 2, that those happen as distinct separate processes. Okay, so how would we draw that? Well, we start with our reactants here. I'm just going to draw them like that. Okay, we start with the reactants there, and we know that our products have to be about the same. So these are going to be the reactants. And we know that our products have to be about the same because of the balance, the fact that they both end up being about -50 kilojoules per mole. Okay? But what we also know is that the intermediate here should be pretty low. Right? Because the first reaction has to be inorganic, and the second reaction has to be, the first reaction has to be exergonic, and the second reaction has to be inorganic. So we know that our reaction is going to end up looking something like this, right? Where we get a reaction like that looks exergonic, and then the second reaction like this is going to look inorganic. Right? So exergonic and inorganic, and this is then I messed that up because I couldn't draw it on the same line. This is then going to be our intermediate. Okay? Now, do you see any problems with this in terms of whether or not this reaction is going to happen? If you put a bunch of reactants in, would you expect to produce products? I see people shaking their head no. Why not? Why wouldn't it happen? Because the second reaction is inorganic, right? And remember I told you at the very beginning of the course that kinetics is something that gets that really gets the short end of the deal in the course, so we don't spend a lot of time talking about kinetics. But I said that one of the things we had to think about was not just whether it was thermodynamically favorable or unfavorable, but the activation energy. Right? Activation energy was really key. And I said that one thing you could do with enzymes is that you could express, the way we could use enzymes to control kinetics is you could express an enzyme, and by expressing an enzyme you could make the activation energy much smaller. Right? So that the thermal energy of the molecule at room temperature was sufficient to overcome the activation energy. Okay? The problem here is that if we look at this activation energy, right, this is the E activation energy, EA, I've defined that for you already. That's the activation energy. It's really large. Okay? This is way too big for this intermediate to have enough thermal energy at room temperature to make that reaction occur spontaneously. Okay, it's not going to happen. So one of the interesting things in thinking about the gap-DH reaction is how does the gap-DH reaction then catalyze this process where both of those processes occur. And the way that it does it is really quite beautiful. And I'm going to draw you another reaction coordinate diagram over here to introduce the way in which it occurs on the gap-DH enzyme. So this is the same type of coordinate diagram that I showed you over there, where we have reaction progress here on the x-axis, and we have free energy on the y-axis. Okay? And much like we had on the left, we're going to have the same situation where our reactants and our products are about the same energy, okay, as shown there. And I should tell you that this is actually the gap-DH reaction. I know that gap-DH is in the shadows there. I hope you can see it. But this is the gap-DH reaction here on the right. And what happens here is that instead of forming this intermediate, okay, instead of forming that intermediate and releasing all that energy, that's exergonic, it releases all that energy, instead of doing that, the enzyme says, I know what we'll do. We'll trap all of this energy. We'll keep it on the enzyme surface so that we can use it and harness it to drive the second reaction. Okay? And the way that that happens is you form an intermediate, and that intermediate is very high energy. The intermediate is very high energy. So what ends up happening is the first reaction looks like over there, but now the exergonic nature of it is a lot less. And then the second reaction, therefore, has a less inorganic nature to it. Okay? The second reaction has a less inorganic nature. And therefore, you can see that the activation energy is a lot less. So let's draw on here that these are the reactants and the products, just so you can keep track that we're doing the same thing over here on the right. Okay? But what happens here is we go by way of a thioester intermediate. We go by way of a thioester intermediate. Okay, so in other words, that first reaction, that first process, the energy is captured and stored, if you will, conserved by forming a thioester intermediate. We've already seen a thioester intermediate. Remember we talked about that in the context of acetyl-CoA? Remember we thought about forming that high energy intermediate, that thioester bond? And now we're going to form a thioester bond here, and that traps the energy, prevents it from diffusing away from the enzyme so that it can be harnessed to drive the second process forward. And that's why we only get a delta G naught prime of a lousy positive 6.3 kilojoules per mole. Okay, so let's take a look at how this actually happens then. Take a look at the mechanism. This is the mechanism. And what we have here, we're going to start the picture here. Okay, this is the starting point of the diagram. I know sometimes that can be confusing. These diagrams all seem to start at different locations. But here's where we're starting the diagram. And notice that we have three key players here in the enzyme active site. So just as always, that hoop there, that blue-green hoop, designates the enzyme binding pocket. That's GAPDH. Okay, we have three key players. We have a base. The base is going to be histidine. Okay, we have a base. The base is going to be histidine. We have a thiol. Okay, maybe not surprising because we've got to form a thioester intermediate. And the thiol is on a cysteine residue. Okay, the thiol is on a cysteine residue. Not so dissimilar from what you've seen already, right? We talked about these types of things already. We showed these thiols in the way that they react previously. Nothing different about this except it's on a cysteine residue inside of that binding pocket. And then we have to have the NAD plus there too. Okay, the NAD plus has to be bound, and that has to happen first. And the reason that the NAD plus is bound and has to be there is because it lowers the cysteine pKa from 8 to 5.5. Okay, the NAD plus lowers the pKa of that H there so that the histidine can pick up the H and so that we can get a reaction there with the first substrate. Okay, so we can get a reaction there with the glyceraldehyde 3-phosphate. Okay, so that's really important to see that the NAD plus is there. Okay, then after we get that, now we've got this intermediate here. That is a thiohemiacetal intermediate. Okay, and that's a name that I want to make sure you know. Okay, thiohemiacetal intermediate you should know, because that's not the thioester intermediate. Okay, we haven't got to the high energy thioester intermediate yet. That is a thiohemiacetal intermediate. Now what we have to do is we have to do the oxidation reaction. Okay, we're latched onto the S, and now the NAD plus does its business. GAPDH catalyzes that redox reaction. We get oxidation, and we go from the aldehyde there to the, we go from the carbon there, which has an H, an R, and an O, to the C double bond and O there. Okay, so that's the next thing that happens. Okay, and now we've got our thioester. Now we've got that acyl thioester intermediate. Okay, and that is our high energy intermediate. And so what we've done is here's the oxidation reaction, okay, that I told you was very favorable, very exergonic. But it's not exergonic here because the product is not this product here, but rather the product is something that still retains most of the free energy. Okay, and the thioester intermediate. Okay, and notice here that that H that was on originally, that was on that glyceraldehyde 3-phosphate, that H gets transferred directly. It's a hydride transfer to the NAD, and that's the exact same thing I drew for you on the board over there. Okay, so it gets transferred, or maybe it was here. Excuse me. Okay, so now we're set up, and now we can do the acyl phosphate formation. Remember that was the second thing we needed to do, the acyl phosphate formation. So now what we do is we do the acyl phosphate formation, but before that happens, we have to swap out the NADH for an NAD+. Okay, the NADH has to be swapped out for the NAD+, and again, that NAD+ turns out plays an important role. And the reason it plays an important role is because it polarizes the substrate here, and it sets it up so that the phosphate has an easier time attacking it. Okay, it drives the reaction by polarizing the substrate. And then that phosphate can attack that carbonyl there, okay, and the histidine gives back its H to the thiol, and we get release of the products in the form of 1,3-bis phosphoglycerate. Okay, so this is the way that the reaction goes, these 5 steps. Okay, and I want you to make sure you understand and know the thiohemiacetal intermediate and the acyl thioester intermediate. So this is kind of a cute little mechanism, but how do we actually know that it happens like this? It seems a little complicated, And it seems like also experimentally it would be very difficult to prove that these two processes were happening on the same enzyme. So let's talk about how we know that it actually occurs this way. So let me tell you about the mechanistic proof that we have that GAPDH follows the mechanism that I showed you on the previous slide. And a couple of these points are going to be old news. Okay, I've already told you that GAPDH transfers a deuterium, an H superscript 2, from the C1 of GAP to NAD+. Okay, we've already said that. One thing you should think about, and you should know how to do now, so go back and try this as an exercise at home for fun, is where would you put the deuterium on glucose to get it at the C1 position of the glyceraldehyde 3-phosphate? Right? We now know how to do that. Where could you put the deuterium on glucose so that you'd have a label there and that C1 position of the glyceraldehyde 3-phosphate? Okay, but if you have a deuterium there, then you can transfer that to NAD+, as I've shown you. The second piece of information, and I only briefly, very briefly mentioned this when we started to introduce glycolysis, is that GAPDH is inactivated by iodoacetate. GAPDH is inactivated by the compound iodoacetate. Okay, so iodoacetate is an inhibitor of GAPDH, and I told you earlier on in this, when we were first introducing glycolysis, and also actually for that matter when we were talking about the TCA cycle, I said one of the ways in which the cycle, the TCA cycle and glycolysis was characterized is by using inhibitors of specific enzymes. And I said that those were really important because in the old days when they were doing this, remember glycolysis was in the early 1900s, the analytical chemistry technologies that they had access to were relatively crude. And so by adding inhibitors, what they could do is it would allow the intermediates to accumulate to much higher concentrations that would allow them to use less sensitive techniques to try to measure them, which is all they had at the time. And iodoacetate was one of those compounds that they used. So let me tell you how iodoacetate works. I'll also tell you that mercury works a very similar way. I'm not going to show it to you, but if you've wondered what mercury poisoning is, it works very similarly. Okay, so what we have here is an enzyme. This is the GAPDH enzyme. I'm representing the open thiol there. Okay, that's the open thiol in the active site. That's the same thiol that forms this thioester intermediate. We're going to add the iodoacetate there, and what ends up being produced is an enzyme that essentially gets alkylated. Okay, this is an alkylation reaction, and what we end up forming here is the carboxymethyl enzyme there, cysteine. Okay, and the way that we can detect this is by taking the GAPDH protein, the enzyme, and hydrolyzing it, so putting it in acid and making all of the amino acids come apart. So pulling off each individual amino acid, and then looking at that cysteine residue and asking if it forms a carboxymethyl cysteine. Okay, that's how we can go about trying to show if this happened or not. Now the last point here is related to the isotope exchange experiment, and we will spend the rest of today talking about the isotope exchange experiment. Okay? The isotope exchange experiment is going to offer proof that GAPDH is a double displacement reaction. The isotope exchange experiment is going to show that GAPDH is a double displacement reaction. Now you're scratching your head wondering what a double displacement reaction is. We haven't talked about double displacement reactions yet. And so I'd like to take this as an opportunity to tell you about two types of biochemical reactions that we're going to study this semester that occur in metabolism. Okay, this may seem casual, like I'm just inserting it in the middle of our glycolysis discussion, but I want to make sure that you understand this slide. This slide will be relevant to every other slide that we do the rest of the semester, so you should make sure that you understand the two reactions here that are listed on this slide. The first type of reaction, which is not GAPDH, is a single displacement reaction. Okay, the first type of biochemical reactions that we're going to study are single displacement reactions. Single displacement reactions are also known as sequential reactions. Can anyone remember where we've seen this expression, sequential reaction, before? And
like a sequential reaction versus a random enzyme reaction.
Okay, so you used a couple words there I'm going to talk about in a second, but I actually wrote on one of your slides sequential reaction. Does anyone remember what slide that was on previously?
It had something to do with, wasn't it like the induced fit model?
Yep, that's exactly where it was. Yep, yeah, that's exactly where it was. It was in the induced fit model of citrate synthase. and which gets to your question, or gets to your point about whether it's being ordered or not, and I'm going to return to that in just a second. So single displacement reactions occur when all of the substrates come in, bind to the enzyme first, before any chemistry happens. Nothing happens until all the people arrive at the party. Party can't start until everybody gets there. Okay, everybody has to be present before anything can happen. Then the reaction occurs and the products are released. Okay, that's the way this happens. Now, the point that was raised, and which is a very good one, there are two types of single displacement reactions or sequential reactions. One is called random order, and one is called the sequential order. Okay, so in the random order, it doesn't matter which reactant gets there first. Okay, everybody has to get there before the party starts, but one person might get there a little earlier than the other, and that's okay. It doesn't really matter. Okay, if we're in an ordered sequential reaction, then the substrates do have to arrive in a defined order. And this is the example of citrate synthase. Remember that we said that in citrate synthase, that it wasn't until oxaloacetate got there that the binding site was going to open for acetyl-CoA. Because we worked so hard to make acetyl-CoA, and the last thing we wanted to do was hydrolyze that thioester bond in acetyl-CoA and not do anything productive with it. We wanted to make sure, if we were going to hydrolyze that bond, that we made citrate. And so we said nothing can happen until oxaloacetate got there, and that was an example of an ordered sequential reaction, which contrasts with a random sequential reaction. Now the other type of biochemical reaction is the double displacement reaction, also known as a ping-pong reaction. And that is going to be the type of reaction that we see in GAP-DH, or glyceraldehyde 3-phosphate dehydrogenase. Okay, it's going to be a double displacement reaction. Now the way that double displacement reactions occur is the first substrate binds to the enzyme first and transfers the functional group. Okay, then it can leave. Okay, and it can do its business. And then the second substrate can come in later and displace whatever that modification was to the enzyme and catalyze the rest of the reaction. Okay, so the substrates do not have to be there simultaneously. They can do action prior to the other substrate getting there. Okay, so these are two different types of biochemical reactions. What I'd like to do now, in the remaining moments, is tell you about how we can distinguish between these two reactions using the isotope exchange experiment. Okay, so one thing that I want you to know how to do is to take any reaction that we've looked at any semester, all throughout the semester. It could be up till now, it could be a future reaction, maybe a reaction you've never even seen before. And I want you to be able to look at that reaction and design an isotope exchange experiment for that reaction and tell me what result you would get if it was a single displacement reaction or what result you would get as a double displacement reaction. Okay, I'm going to tell you what the isotope exchange experiment is. We're going to do one example here for GAP-DH, but you should be able to extrapolate this and do it for others, and of course we can go through that offline in office hours and help sessions and whatnot. Okay, but you ought to be able to do that. Okay, I promise you that this type of question will appear. I've never lied to you before, and I promise to you that this type of reaction will occur on exams and quizzes. You will see this on exams and quizzes. So with that said, in the remaining 7 minutes, let's see if I can work through the isotope exchange experiment for you. Okay, so what I'm going to draw here is much like I showed you before. I'm going to use a radioactively labeled phosphate. Okay, a radioactively labeled phosphate. And I'm going to react that radioactively labeled phosphate with the product of the GAP-DH reaction. Okay, notice that this is the product. This is not the substrate of the reaction. This is the product. And that is going to be the 1,3-bis-phosphoglycerate. Okay, so that's what I'm drawing here. And now you'll notice that both of the phosphoryl groups that are on this molecule, the two phosphates, are not labeled. Okay, there's no radioactive label in either of those phosphates on that molecule. The only radioactive labels on that P there, which is inorganic phosphate, we said could also be orthophosphate PI, however you want to designate it. Okay, but we've got a radioactive label there. Okay, so we're going to put, what we're going to do then, and this is really the key to the reaction, we're going to mix these with the GAP-DH enzyme. Okay, this is the isotope exchange experiment. We're going to take a beaker, we're going to fill it with solution, we're going to put radioactive inorganic phosphate, and we're going to put the product of the reaction, 1,3-bis-phosphoglycerate, with the enzyme. That's all we're putting in there. Okay, there is no NAD+, there's no NADH, okay, it's not present. It's not present. And you can start to appreciate what we mean by this, because if we have a single displacement reaction, we said nothing can happen unless everything was there. And the double displacement reactions, you can start to see what I'm motivating here. We can get reactions even though only parts of the reactants or substrates are present. Okay, so what would you predict to get out of, in this case? What would you predict to detect in this scenario? What do you think we're going to get if we put GAPTH, the 1,3-bisphosphoglycerate, and radioactively labeled inorganic phosphate in a beaker? What do you think is going to happen? Are we going to produce glyceraldehyde 3-phosphate? And it's a little unfair because I just gave you this reaction today, so you haven't had time to look at it offline yet. But do you think we'll produce glyceraldehyde 3-phosphate? How do we get from the phosphoglycerate to the glyceraldehyde 3-phosphate? Remember, the aldehyde to the mixed anhydride species, or the carboxylic acid. That's an oxidation reaction, right? And what is required to do an oxidation reaction? Some kind of cofactor, right? You have to, if you oxidize something, something else has to be reduced. Is inorganic phosphate reduced in GAPTH? No, right? There's no NAD plus and there's no NADH. We're not getting any oxidation here, okay? So I'll draw an analogy to the type of experiment the type of result that we got when we talked about complex 5. Remember when we talked about complex 5 and you were all shocked that when we isolated complex 5 and we added something like the deuterated, the labeled, oxygen-labeled water, that water would fill all of the oxygens in the phosphates? Remember, because we said we were getting rapid exchange? Okay? And that's the same sort of thing we're going to get here. Okay, so what we're going to end up getting here is, of course, we're still going to have the radioactively labeled phosphate, because it's not all going to get used up. Okay, and we will still have we'll get a little non-labeled species too, and more on that in just a second. So that's the first thing we're going to get. That's not interesting. But the thing that is interesting is we're going to get this product here. Okay, so we're going to get a radioactively labeled phosphoryl group that's transferred to our triads. Okay, and here we have the same intermediate there, so I'm drawing the exact same product that I drew over there. Okay, but now the only difference is I've added a label. Okay, I've added a label. So this and this are exactly the same. It's exactly the same, and you go, nothing's happening. Why did you go through all that? You drew the exact same thing on the reactant and product side, and you did nothing. Well, we did do something, right? Because look, we've got a radioactive label in here, and we did not have one here. We have one here. Why? What happened? Well, let's take a quick look here before we end. What happened, I'm going to show you the mechanism. What happened is that we had this product, that's 1,3-bisphosphoglycerate, here it is here, right? 1,3-bisphosphoglycerate. It bound to the enzyme, okay, and when it bound to the enzyme, it lost its phosphoryl group. Right? So it bound to the enzyme, and that phosphoryl group fell off. It formed this species here, but then the phosphoryl group, the phosphate, came back in and pushed it back to the 1,3-bisphosphoglycerate. But when the phosphate added back, it doesn't necessarily have to be the same phosphate that we took off. In fact, it's improbable that it will be, because we've got a whole glass full of the radioactively labeled, and only one of those that came off. So when the phosphate comes back, it's going to be radioactively labeled. And so therefore, in the product, we're going to detect the radioactively labeled version of the 1,3-bisphosphoglycerate. You still detect 1,3-bisphosphoglycerate, but you detect the labeled version. And because you will lose the unlabeled version, you may detect a little of that in the beaker. That's not very interesting. And we're still going to have a lot of the radioactively labeled phosphate because we added a lot into the beaker. Okay? So when I tell you to do the isotope exchange experiment, what I do not want you to do is to tell me that you looked for a labeled enzyme. Okay? The beauty of the isotope exchange experiment is that it's simplicity. It's elegantly simple. If we were to have a lab for this course, which I hope one day we will, this would be one of the experiments that we would do in the lab. I would give you a reaction, and I would ask you to tell me if it's a single displacement or a double displacement by doing this reaction, because it would be pretty easy to collect the product and just ask if it was labeled. Right? That's something that you could easily do and learn if it was a single displacement or a double displacement. So unfortunately, we're out of time today, but I'll see you on Friday and we'll have a quiz.